Hello, my name is Catherine and I'm a researcher at the University of Strathclyde and I am working on biofilms. And I'm going to talk to you about what's in my slime. So microorganisms, like bacteria and fungi, have to protect themselves from the external environment so they can live and grow. Like we build houses to keep us safe, bacteria and fungi do the same. If they didn't produce a biofilm, they wouldn't survive as well because conditions would kill them faster than they could grow. And biofilms are found everywhere you find microorganisms. So basically, a biofilm is a protective structure that microorganisms produce themselves. So a biofilm might have several microorganisms living in it together. As I mentioned, microorganisms include bacteria and fungi, and they can live in the same biofilm. One example is the bacterium Staphylococcus aureus and the fungus Candida albicans. So Staphylococcus aureus and Candida albicans are part of what we call the skin microbiome. And that is a community of microorganisms that we carry with us wherever we go on our skin. And it's these two microorganisms that I'm really interested in. And this is a microscopy picture of that biofilm with Candida albicans and Staphylococcus aureus. The stick-like structures are the fungus, and the little ball-like structures underneath that are the bacteria. And you can see it's like a kind of mesh. So, what is in the slime? So first of all, there's the cells. So biofilms are sticky slimes that the bacteria and fungi make themselves. So the cells also produce proteins, and so they produce them and they go out and into the area around the cells. And they also produce carbohydrates in the form of polysaccharides. Now, polysaccharides is a big term, but it's basically lots and lots of sugar stuck together. And they also have something called extracellular DNA in them. So when a biofilm is forming, some of the cells that are going to make this biofilm pop and they release all their cell contents. And the DNA that's in the cells, so the DNA is the information that cells carry so that they can function. So it basically contains all the information on how a cell works. So the cells pop and they release this DNA into their surroundings. And so when it's out of the cell, it becomes extracellular DNA because it's no longer in the cell. And this is another microscopy image of a biofilm with Candida albicans and Staphylococcus aureus. And you can see these different components. So all these different colours are a different component of the biofilm. So, where are biofilms found and why do you get such a variety of them? So you get a massive variety of biofilms. And that's because there's many, many different surfaces that the microorganisms have to attach to and grow on. Also because biofilms are protective, they have to protect against lots and lots of different conditions. So I'm going to show you some pictures of different biofilms. So here we have biofilms growing on whipped cream. Now I've had coffee cups that kind of look like this. There's a biofilm that you get on your teeth, so dental plaque is a biofilm. And this picture here is of a biofilm that protects its inhabitants from the UV rays of the sun, because unlike us, bacteria and fungi can't put sun cream on, so they have to make their own. So now I've given you examples of different types of biofilms. I'm now going to talk about the ones that are beneficial to us and ones that are problematic. So what are the good biofilms? So biofilms are very important for the environment. For example, biofilms in streams and rivers support these ecosystems by providing food and nutrients for the plants and animals that live there. They're also part of the nitrogen cycle, so that means that we get nitrogen back out of the dead plants and animals and we get it back into the atmosphere, so that's constantly cycling. Biofilms also support plant growth by forming around roots and providing nutrients, and can help protect plants from infection by other bad microorganisms. Biofilms exist in many places around the world and have a variety of important roles in supporting life on Earth. We're used to talking about the impact of climate change on wildlife and plants, but microbes and their important life-supporting roles are also threatened. So what are examples of bad biofilms? Because biofilms are protective, they become a problem to us when we need to get rid of the microorganisms that live in them. Plaque on teeth is a biofilm that contributes to tooth decay. If you eat something sugary, the sugar feeds the bacteria that live in the biofilm, they produce the acid, and that's what leads to tooth decay. Biofilms can also cause infections, and those infections are hard to treat. And they also increase something called antimicrobial resistance, which is what I'm going to talk about next. So what is antimicrobial resistance? When we talk about antimicrobial resistance, we're talking about the ability of microorganisms to survive the treatments we use to get rid of them. So the microorganisms don't die when we treat them with antibiotics or antifungal drugs. This image here shows the growth of bacteria in grey, and where the bacteria has been killed is white, and the lettered discs have antibiotics on them. You can see that 3 of the antibiotics kill the bacteria because there's a ring of white around them, but 1 of them doesn't because it doesn't have a ring of white around it. Antimicrobial resistance arises when microorganisms gain the ability to survive against treatments. You do this by mutation. When this happens, we have to either change the drugs we use on them, or we have to increase the dose of the drugs. And this can also be a problem because if you increase the drug dose, it can start to make the person ill from the drugs. And antimicrobial resistance is a serious global problem like climate change, and we have to tackle it globally. We need new antimicrobials and new treatment methods. So how are biofilms involved in antimicrobial resistance? We know that biofilms are produced by microorganisms to protect themselves. When an infection is caused by a biofilm, the biofilm, because it's protective, reduces the ability of antimicrobial drugs, the immune cells, and disinfectants. It prevents them from reaching the cells. And the conditions inside the biofilm make it more likely that the microorganisms will mutate and become resistant to treatments. And they'll also pass that mutation, that ability to resist the drugs, to the rest of the cells that are in the biofilm, so they spread the ability to survive throughout the cells. Biofilms are very sticky and hard to remove, so if you think about trying to remove a sticker from a surface, so a biofilm infection is really hard to get rid of because of several reasons. Because of these several reasons. So how is my research at Strathclyde helping? So I'm looking at a biofilm that has both Staphylococcus aureus and Candida albicans in it, because that biofilm can cause serious infections. So I'm looking at the structure of it, and we know that how a biofilm protects its inhabitants depends on what it's made of and its shape and structure. So I'm looking at the structure of it and finding out what it looks like and how it reacts to drugs and things. So finding out the structure will help us know how to treat the infection. Because if you know how to build a house, you will know the best way to knock it down. So I'd like to thank Medical Research Scotland and the Daphne Jackson Trust, who fund me and support me, and the University of Strathclyde, and the Hoskinson and McConnell labs, where I'm based at Strathclyde. In my next video, I'm going to show you how to build your own biofilm. Thank you.